Here we introduce a new method to predict regulatory mutation by using whole genome sequencing, DNA methylation, gene expression, and chromosome interaction informations. Here we apply the method in cancer, a lymphoma study. The method mainly have two parts. The first part is a new machine learning method to predict differential methylation based on genome-wide WGBS data. The second part is by combining our predicted differential methylation information with the differential gene expression information and 3D genome structure like high-C data or topological associated domain information. Try to combine all this diverse information together to predict the functional regulatory mutation in the genome and with their putative targetings. And here we look at the method more in zooming figures. For example, our method mainly can look at the data mainly for genome data and epigenomic data. And like we already mentioned about here, is the topological associated domain information and mutation information, gene expression information. And we also combine this kind of diverse genomic data with a biophysical model of a protein DNA interaction to predict mutation changes in the non-regulatory region, in the regulatory region. And here there are mainly three packages like base pair bar, basing, bounding affinity ranking method to combine the mutation block and the differential expression information. And another method called DMR analysis, the differential methylation analysis pipeline to find the genome-wide methylation pattern based on whole genome methylation data. Then based all the information combined together with another integral method, DDS method, by combining all the differential methylation, differential expression, gene expression, and the mutation information and the mutation frequency across patient, try to predict the functional mutation block which are maybe affect transgenic binding and the targeting dysregulation in the cancers. Here is the pipeline, the cartoon figure. Now we go to more specific each part of the work. Here, first talking about differential methylation. The new differential methylation analysis is based on the WGBS data. It's a quite large data set. Here, we first identify the methylation region genome widely. Then we base on 6 features in each methylated region, try to make a classification. First, we have like have a principal component project methylation changes and a differential methylation CG size changes and all kind of future scores produced within each methylated regions. Then we use logic regression model to try to classify the this kind of methylated region into differential methylated and non-differential methylated regions. Finally, we using some ranking score to rank the predicted differential methylated regions. And this kind of information will be used later on in the second part of the work by combined with the mutation information. For example, here we have the SNP, single nucleotide variant from the genome-wide sequencing data from patient. Here the lymphoma patient, follicular lymphoma. And then the mutation block is predicted by base pair bar 2. And here the mutation block information will combine with differential methylation information. And also that they are relevant genes, if for example this mutation block linked to the gene either from the promoter region or the 5' distant region. And all kind of putative targeting should be locally same topological domain. Here we also develop another score system with the voting system, try to predict which mutation block are possibly functional in the regulatory region. The information mainly consider like, for example, frequency, number of patients that contain these mutation blocks. Then a differential methylation score, for example, how far these mutation blocks are overlapping with differential methylation and how strong this differential methylation is based on our prediction. Then is the differential express genes linked to these particular mutation blocks. Finally, the received mutation block in the located in either gene or enhancer or fine distant region assign a weight. Finally, based on all this kind of information, a weight vote system will predict which mutation block are strongly affect downregulate downstream gene difference between the tumor normal samples. And then we can predict top ranked gene mutation score associations. For example, one example from our prediction top one is the BCL2 genes. And here we look BCL2 gene here in these figures. Red color, that's the mutation block, differential mutation block predicted by current method. Light blue color, that's the enhancer region from the database. Dark blue color, that's the mutation blocks predicted from genome data. Here the black line, that's the topological associated domain. So based on our prediction, BCL2 ranked on top 10 list, and they are associated. It's this mutation block overlapping with differential methylated regions in the near to BCL2 genes in their non-coding region. And if we look at this differential methylated region, MR2256, MR22253, you will see red color, green color represent tumor and the normal samples. They are very clearly till differential methylation happens overlapping with differential with the mutation blocks, and this mutation block possibly affect the one important gene, the follicular lymphoma BCL2. Okay, more information about this work can be will be available in our online website and publication here.